DNA fingerprinting is what I'm going to talk about, also known as genetic profiling. I'm going to start out by giving you a scenario and letting you know like what I came up with. So Jesse Smith and his girlfriend, Sue Marsh, stopped on their way home from a late-night movie to make a withdrawal from an ATM. Sue stayed in the car while Jesse walked up to the machine. As soon as he punched in his PIN number, a man in a ski mask swung a tire iron at Jesse. Jesse was brutally beaten by the attacker. He fought back and in the process clawed the attacker. The attacker took the money and fled. Jesse was taken to the hospital and later died from his injuries. Sue was able to identify two likely suspects after viewing mugshots at the police station. The police took the two suspects into custody, but both had an alibi. One suspect had a scratch on his face, which he claimed he got from his girlfriend. The police recovered samples of blood and skin from underneath Jesse's fingernails and also took DNA samples from both suspects and sent them to the lab. You are the lab technician on the night shift at the forensic lab. You received all the samples of DNA collected as evidence. Your job is to find out whose DNA was under Jesse's blood and which suspect it belongs to. So today I'm going to give you a recipe in extracting nuclear DNA and the process of electrophoresis. Both applications are used in identifying a person. First is extracting nuclear DNA. This was from some of the stuff that I did, actually. And I don't know, did that come right? No. Can you see it?
Adjust it a little bit, maybe.
Where do you adjust that? Oh. There you go. So first, you're going to get a small 15 milliliter tube, pour a small amount of solution with electrolytes and the DNA sample from a suspect into the 15 milliliter tube. Place the tube on a test rack, or test tube rack, and let it sit undisturbed for 30 minutes. After allowing 30 minutes, add 2 milliliters of cell lysis to the solution. Cap the tube and invert 5 times. Allow the solution to stand for 2 minutes. After 2 minutes, hold the tube at a 45-degree angle and carefully insert 70% ethanol into the mixer using a plastic pipette. Then let stand for another 10 minutes. During this time, wispy strands of translucent DNA begin to clump together where the ethanol meets the cell lysate layer. You now use a pipette to transfer your DNA out of the 15 milliliter tube and into a small container. The DNA, by the way, is extremely brittle, and if you try to use the pipette, a lot of times, it'll break, so you have to be extremely careful when you use it. You're going to add a little bit of loading dye to the DNA, and now it's ready for use in the electrophoresis process. So the electrophoresis process is kind of where you're going to read your samples of DNA. So the next step is the electrophoresis process. Heat some agarose gel to 50 to 60 degrees Celsius. Pour the gel into a gel box that has a comb in it. This is some of the other work that I did. Wait until the gel comb cools, or the gel box cools, and remove the gel comb, leaving a row of wells in one end of the gel. Now pour a TBE buffer solution, 3 millimeter depth, over the gel. Using a syringe with an adapter, which I kind of like this, you're going to extract one at a time each sample of DNA from the small containers and insert them into their own separate wells in the gel. Next, cut 2 carbon fiber copies, fiber tissues, 42 millimeters by 22 millimeters, and insert them on each side of the gel box. So it's kind of like what I did here. I don't know if you can see that fiber. If you look at the box on number 2, you can see that it has fiber paper inserted. I don't know if you guys can see that. Can you see the fiber? There's like black paper on each end. And so you're going to cut the fiber, you're going to put it there. Then you're going to use electrodes and attach 3 9-volt batteries to the end of the gel box where the wells are, making sure the negative charge is next to the DNA wells and the positive charge is on the opposite end. The DNA molecules are negatively charged and they will respond to the electric field and migrate toward the positive electrodes because of the molecules of the DNA. Bigger molecules tend to stay closer to the negative charge, and the smaller molecules tend to stay, further, go toward the positive charge. The DNA molecules, I will leave the gel to run undisturbed for several hours. When the loading die has reached the end of the gel, disconnect the electrodes, pour off the buffer solution, then pour about 10 millimeters of Carolina blue concentrate onto the surface of the gel and leave for 4 minutes. Rinse the gel with cold water about 4 times. This is what I came up with. Put the gel in a plastic bag and leave to develop overnight. The next day, place the gel on a white background so you can see the DNA bands that show up vividly, and compare the evidence DNA to the evidence from the 2 suspects. To compare the bands of DNA, we look at where the final locations of the bands rested and how many bands are in each row. If the amount of bands and the location of the bands are identical, then they are the same DNA. In conclusion, after comparing DNA evidence, we positively identified the suspect with a scratch on his face as the attacker. We have gone from the crime scene to the steps of extracting DNA to processing DNA and coming up with usable information to accurately identify a suspect in the crime. I hope this information has helped you understand the DNA fingerprinting process a little better. I'm nervous. I totally had to read. Should I move it back?
Should be fine.
Yeah.
You guys learn anything as you guys all shoot me on it. No. I was looking right at you while I'm talking, and you're on.
No. I did learn. I had a long night. I'm sorry.
At least it's Is it? Oh, I feel like you're part of it.
I thought I did.
Oh, I'm so glad
Congratulations, you
I look so forward.
I know.
I heard you when I tried to read it to her, literally like a recipe. And she was like,
And what would it
well, that's interesting. Okay.
You did good. You look nice.
I look nice? Really? Even though it's like that.
She went to the...
Like I knew
I
that.
know what you're talking
And I
about.
did biology and chemistry that day.
All right, Gigi, I think what you need to do is start with the example, because you just basically start with the topic first. But if you start with the example, that'll pull us in, and then you can identify the topic. You've just got those 2 things backwards in the introduction. Your goal is pretty clearly stated. The content is okay. It's a little dry sometimes, and it sounds like you're giving us a recipe, and you're giving us a bunch of materials that we're not ever going to be able to see or do. So I think that you need to simplify some things and explain, put it into basic steps instead of telling us every ingredient that you add to the gel and the number of times that you turn it over, the angle that you hold it at, which, I'm sure it's important when you're doing it, but I would be incredibly surprised if anybody in here ever needs to do that. So it's just one of those things that I think you are giving us the wrong kinds of details. I think you need to pick some other information from the process to emphasize for this audience. On the presentation issues, you are kind of limited in that one spot. Your gestures are limited because you've got the script in your hand the whole time. You've got a whole script, and you tend to hold it with both hands most of the time. There are once in a while a few indicators. I thought you were expressive when you were talking to us for the most part, not in a wide range, but so that we could tell that you are in fact engaged in the subject. Your eye contact is really inconsistent because you tend to focus on the notes that you have in your hand and the visuals when you're making reference to them. When you do look up at the audience, you're looking kind of in the center of the room. I know, once again, the camera and myself become a focus of attention. Don't forget the people on the sides of the room. They're part of your audience too, and you need to be engaged with them a little bit more. The strongest parts of your presentation, I think, are your voice. It's easy to hear you. There's good variety in your voice. Sometimes the pacing is a little bit awkward, especially when you start reading through some of the steps that you're going through. That seems a bit awkward. Anxiety control, I would have said a little bit better, except right at the end you just, when I said earlier to reduce your anxiety that you ought to share it, I think you should do that before your speech or after you sit down, not while you're standing up there speaking to everybody. I think that creates a little bit of a problem. Okay, thank you.